my name is Hassan. I'm going to be going over prompt 41B with point mutations. So a point mutation results in the substitution of one base pair for another. There's a transition mutation where a purine base is switched for another purine base, or a base switched for a G, and a pyrimidine base is switched for another pyrimidine base, or a C base is switched for the T. And there's a transversion when a purine base is switched with a pyrimidine base. We're going to be using this diagram to figure out whether the mutation was transition or transversion. Alright, so we are going to be using the given non-mutated original sequence to get to our transcribed sequence. And once we have that, we can translate it to the mRNA sequence, and that is used to determine the corresponding amino acids. To our first example, a missense mutation is when the change of a single base pair causes the substitution of a different amino acid in the resulting protein. A missense mutation can also be neutral, meaning it won't affect the protein's functions. Here we can compare our original DNA sequence to our mutated sequence, and we can see that an A base has been switched with a T, meaning that transversion has occurred. Comparing the amino acids, the original DNA sequence gave us the amino acid asparagine down here, and the mutated DNA sequence gives us the amino acid tyrosine. Once again, meaning that missense mutation has occurred because there has been a substitution of a different amino acid. Next, we have a nonsense mutation. A nonsense mutation changes the codon, so it becomes a stop codon. While comparing our original non-mutated DNA sequence to the mutated sequence, we can see that a G base has been switched with the A base, causing a transition mutation. Looking at the amino acids, the original DNA coding sequence contained a tryptophan amino acid. However, due to the mutation, it becomes a stop codon, once again meaning that nonsense has occurred. You can see that down here. A silent mutation is a change in the nucleotide sequence of a gene that does not alter the amino acid sequence of the encoded protein. So comparing our original non-mutated sequence to our mutated one, we see that a C base has been switched to a T base, meaning that transition mutation took place once again. Looking at the amino acid sequence from the original DNA coding strand, we have a cysteine amino acid, which is the same amino acid we get from the mutated strand, so they did not change. A read-through mutation is when a stop codon is changed to a codon that codes for an amino acid, resulting in a longer protein. So comparing our original sequence with a mutated sequence, you can see that a base has been switched for a G, meaning that once again transition has occurred. Comparing our original amino acid sequence to our mutated one, we can see that the non-mutated sequence resulted in a stop codon, while our mutated strand translates to a tryptophan amino acid, which also results in a longer sequence of amino acids.